Hello, everyone, and thank you for joining us. I am Lori Day. I apologize for the delay in starting this. We have some technical difficulties, but welcome to our second day of the second virtual Lupus Mini Health Conference presented by the Lupus Foundation of Northern California. For those of you who attended yesterday, this introduction will sound a little familiar, but I'm repeating it for anyone who didn't attend or attending for the first time today. My name is Lori Day, and I'm excited to be your host today. I began volunteering with the LFNC in 2019 and then started working part-time with them shortly thereafter. I've really enjoyed meeting lupus patients and working with them over the last 2 years, and so I'm very excited to be moderating here today. But it is only my second time, so bear with me if I sound nervous or I mess up. But before we start with Dr. Niewold, a few housekeeping items. The Lupus Foundation of Northern California has one more health conference planned, and that will be a Spanish language one on October 16th. We also have our Outrun Lupus 5K happening on September 25th. And this will be a hybrid event, so if you live locally, you can join us at the Campbell Park in Campbell in person, or you can join virtually wherever you are. Feel free to check out all the details at outrunlupus.com and invite your friends and family. For additional information about all of the different programs we have, you can go to our website, www.lfnc.org, or you can email us at communications@lfnc.org. So we hope some of you were able to join us yesterday for Dr. Balboni's presentation on lupus in children, as well as the wellness session with Julie and Kat. But if you missed them, the recordings will be available on demand next week. You'll receive an email with the link to them. So now I'll go over the agenda quickly and we'll get started. The first hour, Dr. Niewold will discuss the genetics of lupus, and afterwards he'll be answering some questions. Any question that you might have, you can ask by clicking on your Q&A button in the toolbar of your Zoom window and typing your question. You can do this either during the presentation or after during the Q&A session. You can also ask questions anonymously by clicking the checkbox next to your question before clicking submit. So don't be shy. We will have a break and then return at 1 o'clock to join Regan Burr as she talks about her lupus journey. She will also lead us through some stretches, so grab a towel and some water for that session. Remember, you can attend any of these sessions using the same link. So if that sounds interesting and you didn't sign up for it, don't worry, just click the same link and join us. And before you leave, be sure to complete our quick survey. It looks like we have about 19 people signed in right now, and I'm sure we'll have more joining us. But let me introduce Dr. Niewold. Dr. Timothy Niewold is the Judith and Stewart Colton Professor of Medicine and Pathology at New York University School of Medicine. He's also a physician scientist and is the director of the Colton Center for Autoimmunity, which focuses on the identification of causal factors and new treatments targets in autoimmune disease. Dr. Niewold is a human geneticist and translational researcher. He bridges the traditional gap between the basic and clinical sciences. Work in his laboratory focuses on identifying and understanding the pathogenic factors in human autoimmune disease and the ways in which underlying genetic risk factors impact immune responses to result in disease. Welcome, Dr. Niewold.
Great. Thank you. Thank you for having me. The technical delay was really on my side, and I apologize to everyone about that. I had my computer on and working, and everything's working well, and then I go to jump on the Zoom 10 minutes before, and it took me a lot longer than 10 minutes to get up and running again. My internet crashed. Anyway, I'm very sorry about that, but thank you all for staying on the line, and I hope that this will be interesting and exciting for you. So I will do the share screen now. Okay, let me get to the slideshow mode here. Okay, it's taking a minute on my side. Can you see the slides now?
Yes, looks great.
Yes. Okay. Okay, thank you. Okay, so thank you for the kind introduction. As mentioned, but we'll talk about genetics, and I'll try to, we'll do some general genetics just to sort of bring everybody up to speed a little bit, and then I'll try to bring it into where we are, though, in thinking about lupus, and even how genetics has in some ways informed some of our new treatments that are coming online this year. So I think it's really an exciting time in lupus in general, and I think that it's a nice example in the interferon pathway how genetics has actually led us through all the way now, as we're all excited about, to a new FDA-approved treatment. So I think it's really a nice time. I'll sort of go back to basics and talk a little bit about genetics, and then we'll go through how genetics impacts the interferon pathway, and then talk about how this might be useful even as we think about a new potential treatment coming to us that we can use in the clinic. So, all right. So lupus, I know the audience is very familiar with lupus. This is just my sort of set point slide, thinking about lupus. It's sometimes called the prototype autoimmune disease. You may have heard that, but I think if it is the prototype, it's still got some fairly mysterious disease manifestations, and I think it often arises differently in different people. I can say from the physician side of me, you see lupus do lots of different things in different people. I've often said that if you had 4 lupus patients in the waiting room talking to each other about what's happened to them in their medical life, and they couldn't use the word lupus, they might not think they have the same disease even. It can be very different in different people. The other challenge really is this variable and unpredictable course with players in remission, and our tools to deal with that are not great. We have nonspecific immunosuppression. Many of you who are patients in the audience will have experienced hopefully some benefit from treatment, but unfortunately, many of you may have experienced some side effects as well, because if you've had a life with lupus, it's not uncommon that you may have some side effects from some of the medications you've had to take from time to time. So we really need to understand what causes disease and how it happens, I think, to really direct our therapeutic efforts and to be more specific. I think that's the path forward. How can we be more graceful with our treatments and really just affect the lupus part without affecting other parts of the body and our normal immune system? I think we really have to understand better how everything gets started. And I'll say, as a physician researcher, that's one of the things that drew me to genetics, is I think that that's one of the ways you can get back to some of the root causes of disease. So pictures of lupus, again, many of you may have seen these or had these manifestations before. The classic malar rash often affects the cheeks somewhat symmetrically. It doesn't have to be perfect, but there's some symmetry to it, crosses the bridge of the nose typically, spares the nasolabial fold, which is interesting. If you draw a line from the edge of the nostril down to the corner of the mouth, usually that area is spared. And I say that because it's an interesting thing we've known in the history of medicine. That's the way the rash comes, and it's striking. If you ask me, from a molecular perspective or, an immunology perspective, why does that happen? We don't know. There's something, it's interesting, right? So it happens, it definitely happens. If you wanted to get down to the actual molecular reason, we don't really know. So there's still a lot of mystery, I think, in lupus that we're trying to unravel. The slide images are pictures of kidney inflammation under the microscope, both the fluorescent green stain and then the tissue stain next to that, and then some of the pictures of the rash in between the knuckles and the arthritis that can happen. And so really challenging disease, as probably all of you know, that can affect so many different body systems and at different times in different ways in different people. So maybe I don't even need this slide after saying that, all the things I've said on the first two, but I think we believe this, but why? Why is lupus heterogeneous? And I think this is a challenge, this last bullet, this is a challenge for anyone, I think, on the academic side like myself that wants to help move the field forward, or if you're working in a pharmaceutical company trying to develop drugs. I think this is a challenge for all of us, is can we do something besides one size fits all, and can we try to, understand the heterogeneity of lupus such that we could intervene in a personalized way for individual patients. And that's really, I think, a goal that we're pushing toward. I don't think we're there right now, obviously, but I hope that we can get there. So, so precision medicine, this is a term you may have heard, and it's a popular term now. I think many times when people use this, they're thinking about either genetics or other what people call biomarkers, or things you might test in the blood that might identify a different subset of disease or a different group of patients that might need, different treatments than another group of patients. And so one thing that I think is funny is if you're in medicine long enough, then now we say we're doing precision medicine. I feel like older physicians might feel like the implied other part of that maybe they were doing imprecise medicine before. I don't think so. I think every generation tries to do the best with what they've got, but I think the term is trying to imply that there may be more things we can bring to the table now, and that's our hope, is that we can bring some of this new information to the clinic and apply more information than we used to be able to, and hopefully this curve will continue. If we're lucky that this will continue, that 20 years from now that we'll be doing things we never would have dreamed of now, and they'll look back at what we did now and say, wow, I remember when we used to do it that way. So hopefully progress will continue, but this is one of the latest terms that's been used quite a bit for this idea of applying genetics or genomics in medicine. And so why does this happen? So we are both the same and different. This is a slide that I actually took from Scientific American. They had a nice article a while back about human genetic variation. And so both things are true. We're very similar to each other in that we're a pretty new species on planet Earth. We've done well. I think it's safe to say we've just about conquered the whole landscape, so that's pretty good in the short period of time we've been around. But we have had different populations living on different continents, and we've had some chance to evolve somewhat in different ways, or have drift happen to our chromosomes in different ways. So people living in sub-Saharan Africa didn't have so much contact with European populations for a long period in human history, and so there are also differences. We're probably not as dissimilar from each other as Darwin's finches, but we have some differences that have arisen both within and between different world populations. And so there have been some really fascinating studies recently looking at this. You can see that population genetics actually seems to correspond to historical data like archaeological finds and things like that. And so we're sort of piecing together human history from these different sources. And you can track, genetics and Y chromosomes through human settlement. We all began in Africa and then have spread across the globe, and you can see this in our genes, actually. So if you trace back, different human genetic variations, you can see different arms of human populations that have gone and settled in different places. Some of the most recent being Native American populations settling in the Americas, and that story happens, between 20 and 10,000 years ago. Some of the ways we think about genetics most commonly, I just want to define a couple terms before we go forward and talk about these. Because we're pretty similar to each other, the most common human variations, or many of them, are single nucleotide polymorphisms. So that's, if you think about the DNA code, the ACTGs, that if you lined up all the letters, the four bases on our genetic code, this would mean that in one specific spot in the millions of letters that make up our DNA bases, there's a change maybe from an A to a T or a C to a G. And so that's pretty common throughout the genome, and you can think of that as a relatively minimal change, right? So the rest of the code in the nearby region would probably be the same between individuals, but then thered be one spot where there's a single base change. They can be anywhere in the gene, so they can be in the part that encodes the proteins, which are the machinery of the cells that then work and do all the cellular functions. They can be in the other part of the genome. You may have heard that only, it's a small percentage of our genome that actually encodes the proteins that make up the cells. And sometimes people had used the word, is the rest junk? It's not junk, it's there for a reason. And then there are a lot of things that the genome does besides make protein. These polymorphisms can be anywhere. So they can be, and they can actually change the way the protein is made and encoded. Those are called protein coding changes, or changes in the amino acid sequence of proteins. Or it can be in areas around the gene that maybe don't change how the actual protein is built or constructed, but it changes the way that it's expressed. And maybe you would make more or less of that protein because there are other genetic variations nearby the gene. So it can, Mother Nature is very clever, and things that happen in the genome happen in all different kinds of ways, and some of these things actually do change biology. So this is how we start to think about how you go from changes in the genome toward changes in physical features, phenotypes like diseases, things like that, is that eventually these things change protein and change biology. There are international projects and consortia that have really mapped this out. If you think about the Human Genome Project, now a lot of the younger students that I teach, that's like ancient history, but I was alive when that happened. That seemed like a pretty big deal to sequence the human genome. Just for reference, that was trying to sequence one genome, one person basically. And so the DNA from that person was sent to different spots around the world, and everybody sequenced parts of it, and they put it all together. So that was a big advance, but now we've gotten to where we can sequence people pretty rapidly, actually. And this is something that we're even wondering whether this could come into medical usage. We do some sequencing of just the gene regions, so the regions that code protein. That's actually done in the clinic now in some cases for rare diseases. And then doing the whole genome, which would include everything, is I think one of the next frontiers people wonder about. So there's this project on the slide where they mapped human variation. The Human Genome Project wasn't anything about variation. It was just one person, so you don't know anything about variation. But there have been subsequent projects after that have mapped common human variations, and it's something that is a useful reference when we do genetic studies. So it's an exciting time. I think we're getting a good catalog. I guess the point I wanted to make is that there's a pretty good catalog of common human genetic variations. And so we're really able to ask these questions about, are there common human variations that lead to risk of diseases? So I just want to talk a little bit about this issue of populations again. So we have to be careful of this in genetic studies, and I think maybe I won't spend a ton of time on this describing study design, but it's just something that we have to be careful of. You could imagine if you wanted to study the genetics of lupus, and you say, I want to take my lupus patients from one country, I'll take lupus patients from Iceland, and then I'll take all my control subjects from sub-Saharan Africa, and then I'll compare their genomes. You'll find a lot of differences, but a lot of those differences you find may relate to just the different populations and their own population history, and not so much anything about lupus. And so you have to be careful with that, because that can happen on a smaller scale. For example, within Europe, there are actually differences in some genes in northern versus southern Europe, and they can be pretty significant. They can be sometimes a twofold or more difference in the frequency of a gene. So you have to be fairly cautious with this when you do these studies. So it's one of the things we have to control for. I thought it might be fun to present just because maybe some of you have done things with like some of the commercial providers that will do personal genetic services like Ancestry or 23andMe or things like that. If you got in your readout, they'll actually often place you, for example, within the European continent. They would presume that there's so much ancestry from these different countries, and you really can get that specific. This is a study that I think preceded some of that granularity and helped show what you can do with this type of data. And this is a British Isles genetic study where they looked within England and the UK. And the different color dots really designate major different genetic groups within England and also within Scotland and Northern Ireland. And you can see that the different colors kind of segregate geographically, with the red actually indicating the areas that were largely conquered and settled by the Romans. The Celtic fringe not so conquered and settled by the Romans. And then there's a line between England and Scotland, actually, where there's a difference, where you see the white and yellow and orange dots, and it's interesting. It's around Hadrian's Wall and the historical divide between England and Scotland, and shows that these populations were somewhat distinct as well. So you can really get a lot of detail out of your genome, and so this was an interesting study. They had done a lot of genetics in the normal population in England, and then they were able to show that you could actually see a lot of population history. The same paper, they went through some of these details about where different peoples had settled on the islands, and so in the color codes there you can see kind of going from the upper left and then down to the lower right on the screen, the different settlements through the years, and so you can actually trace this genetically. So so there's a lot of power there. I go through that because some of you may have had this experience in commercial genetic services where you can actually get a lot of detail, and so you can imagine with that level of detail about ancestry, there's probably something about disease that you can tell as well. So this is a question I like to address because I think it's a question that comes up a lot, is why would my own genes give me an autoimmune disease, though, right? So it's a fair question, because you would think if there's a common gene that causes an autoimmune disease, maybe that would start to be less common over time. Things that cause problems for us. One of the principles of evolution and selection is that if something causes a species or an organism a significant enough problem, it may tend to get weeded out over time. But it's really probably because autoimmune conditions haven't been the major problem for human populations through the years. I think infectious disease, I can say that when I gave this slide before COVID, it was maybe a bigger ask of people to consider how infectious disease has been, a big problem, but it really has. We even before COVID, we lost lots of people to influenza, even in the West and in areas where there's a lot of modernized medicine. You can imagine times in human history like the plague and other times where there were big, death events in the human population related to infectious pathogens. And so, and then if you think even farther back, when we were mostly living in small bands and tribes across the planet, you could imagine an infection could come through and really wipe us out, and there's no doctor, there's no medicine, there's nothing you can do about it. So a lot of our genetic history actually goes back that far. Our recent modern, few hundred years of human history where we've had all these modern conveniences is pretty short in the human existence on the planet. So so a lot of these things probably are that we think are risk factors for autoimmune disease. They could actually be things that have helped us fight infection in the past. You can imagine that there's sort of an arms race, and I think this is how it goes, actually. The pathogens get quote unquote smarter. They find ways to evade our immune system, and our immune system has to get more aggressive to try to contain and control them. And this has been going on, for all of human history, for sure. Once you generate this hardware, once you evolve to have a more pro-inflammatory polymorphism in one of your genes, then these genes are transmitted to subsequent generations in a chance fashion. And so, because you're just as likely to get a chromosome from mom or dad, in inheritance. And we think that autoimmunity likely represents sort of unlucky combinations of genes that come together. Individually, these genes are not quote unquote bad genes, but that you may get, a way I like to put it is maybe you got too much of a good thing. In lupus, we think the antiviral system, including interferon, is overactive. We do need our interferon system to fight off viruses and things, but it might be that you got a few too many of these things that turn on the pathway and make it hard to control, and then that later on leads to risk of disease. So I think that's the principle we're operating with, because many of the genetic polymorphisms are actually pretty common. If you ask, which genes are linked to lupus, some of them are 30% in the population or more. And then, again, you can understandably have some difficulty with that concept. You think, okay, 30% of people don't have lupus, but you say this risk factor is present in 30%, and so the risk factors are all relative risk factors for the most part. But we think that then they come together in combinations and end up causing risk of disease. So I got ahead of myself maybe a little bit, because I think that's what this slide is about. But there are some conditions, so not lupus in most cases, but most of lupus we think of as polygenic. But there are some conditions in rheumatology that are probably due to single major gene mutations, and so these you can track through families with, if you remember your genetics from, sometimes dominant or recessive inheritance, and there's one gene and you can kind of track it through the family. There are some inflammatory conditions like that, but they tend to be rare. And the most of the common things that we see, for example, lupus patient in the clinic, rheumatoid arthritis, type 1 diabetes, all seem to be mostly polygenic cases. Now I say mostly because there are some single gene causes of lupus. Our most famous one that's been known for probably 30 years or more now is a recessive deficiency of the complement C1q gene. It's very rare. if you had a few hundred lupus patients and you sequenced their genome at this region, sequenced all of them, you may have one case or 0 cases. And so it's very uncommon, but there are some people who have some single gene defects that can lead to lupus. But in large part, it's a polygenic inheritance. And for most family histories of patients with lupus, and if you have lupus in your own family, it's actually pretty common to not have other people affected by lupus, or maybe one other person, but it doesn't tend to track through the families in that way, that we learn in Mendelian genetics of dominant, recessive, and things like that. So we've known that for quite a while. The presumption just from our family history studies would have told us that polygenic is probably going to be the answer, and this has panned out. I show this because I think maybe that last example is a good segue to this. This is one of my favorite genetics papers, actually, and it's from 1918. So I don't read a lot of scientific literature from 1918 because a lot of it is not so relevant anymore. But in this case, a lot of the theory and the math was figured out quite a while ago. And so this paper described this idea that if you have multiple genes that can all impact the same outcome, and whatever it is, genes impact things like human height. There were geneticists studying human height in families, and they realized that human height was not a yes-no, that it was sort of there was a correlation between parents and children, but it was not a one-to-one thing. And you could have tall parents with a short child. You could have the opposite. On average, if there were tall parents and they had like 10 children, on average, the children will probably be taller than average, but it was not a simple relationship. And so this sort of more mathematical paper made the point that if you have, say, maybe a way to explain it, if you had like 6 tall genes running in the family, the odds of each child getting all 6 tall genes would not be that great, actually. You could almost picture it like 6 flips of a coin, and you'd have to have all 6 come up heads to get all 6 tall genes. It would happen sometimes, and then that would be a tall child. Most likely, you would maybe flip 3 heads and 3 tails, and you'd be somewhere in the middle. And so, another family where they only had 2 tall genes, you'd probably still come out taller than the average person in that family. But just to give you an idea of how these things can come together. So neither hypothesis was wrong. I think this paper was really strong in that it could unify the old studies of Mendel, where you had this, single gene makes the pea either green or yellow or whatever, and then how can you reconcile this with many of the other things we see in human families? So again, like many things in science, you could come up with a theory and then it can take a long time to prove it. It's been about 100 years from that paper, and we've started to have some really good genetic studies of complex diseases where we've been able to do them well enough, map the genome well enough that you could find some of these common polygenic risk factors that are enriched in people with disease. So this is a picture from one of the seminal lupus genome-wide association studies. So that's before I just use that word and not explain it, genome-wide association just means that it was a study where you could look across all the chromosomes and ask what is most different between people who have lupus versus people who don't have lupus. And so that's really the study design here. And the colorful graph in the middle is just showing the different chromosomes color-coded, and where you see these dots that rise up above the noise there with the labels like HLA and the other labels. Those are gene names, and those are statistical signals that are significant, and they rise above the background noise in the study. And so those are genes where there's an association with lupus, where there's a particular variation there that is associated with lupus. So we've actually been able to find a number of things. That's been a success. During my career, this has been a big success. We went from hoping one day we'd find the genes that cause lupus to actually being able to generate a pretty solid list of genes. So here's a list that's actually out of date by a while, but it's one that can fit on the screen. When I try to show all of our current associations, there are more than 40, and it actually is a little bit hard to show without getting really small fonts. So the gene names, obviously this is just to sort of take it all in, not to read every individual thing on the slide, but the overview is that in the column where it says potential function, we don't always know how all these genes work. They just come out of a screen and that's it. And then if someone's already studied it, we may know what that gene does. If no one's ever studied that gene, then we may not know. But I think it fit our ideas about what causes lupus because many of these genes we knew already had immune system function. And we've always thought of lupus as an autoimmune disease. This is just another piece of evidence that supports that, is that the genetic factors related to lupus tend to be in or near immune system genes. The odds ratio column I'd also point you to just briefly is that these are the odds ratios you'll typically get in a polygenic disease. You could have a particular variation that if you knew that variation was a risk factor and you carried it, that your risk could go up, the top one there, HLA, 2.4-fold. So that would equate to like a 2.4-fold increased risk of lupus. To put that in perspective, the background risk of lupus in the general population is somewhere around 0.1 or 0.2%. To make the math easier, if we said 0.1%, that would, for you, if you genotyped your HLA and you didn't have lupus and you were worried because you had the risk factor, you said, oh, now what's my risk now? It would go from 0.1 to 0.24%. And so that would still mean you go from 99.9% not going to get lupus to like 99.76% not going to get lupus. So unfortunately, I wanted to go through that on this slide because that's unfortunately where we are, I'd say, right now as far as being able to predict lupus genetically. We still don't quite have enough information. I'll give some more reasons why and how I think we can actually make progress on that front. But if you just know these individual things, then even if you put all these individual things together, we're still not quite at a point where we can use this in the clinic to predict someone's risk of disease. But I think there's hope for that, and we've been making some progress in my own group on that question. This one might be a little complicated, and I'll just try to summarize that. Maybe it's best to use some of the lines I used earlier, is that there are many, many different ways you can adjust the function of a gene. So this is a commonly used way we diagram genes, where the black line is supposed to indicate the chromosome, and then we put these blue boxes where there are regions of the gene that encode the protein, which we think is obviously one of the main functional parts of the gene, is that you make these proteins from genes, and then the proteins make up the machinery of the cell. So you can have variations that are right in the blue boxes, but you can also have a bunch that are nearby and performing other functions. So it can change how these blue boxes come together. When you assemble the protein, you assemble the message that leads the protein into these different boxes. You can picture them almost coming together. And when you do that, there can be things that are cut out or put in. And so sometimes the genetic code varies in that way, and all the individual boxes are the same, but how they're put together differs. So Mother Nature is very clever in how many different ways you can change function, and so that does make a challenge for us when we try to find which gene causes the disease and how does it do it. That's after we've generated this big list, it's really exciting to know where some of the genetic variations are, but then we have the hard work of trying to understand how they function. And I'll say that our field generally is at that point now. And this slide summarizes some of the success stories where we have these gene names, in brown, like IRF8, and then we know that there's a particular function, near the end of that gene that alters something in the intergenic region. So we're getting better, but there's still a good amount of work here. The reason I think it's important and why I talk about it is that that's, it's sort of a stepwise way to go, where you find the genes that are linked to lupus. You don't know what they do yet, but then if you can understand what they do, you keep getting closer to that big picture question of what is it that causes lupus and what goes wrong in the first place. Okay. And then this is fairly, I promise we'll get a lot less detailed slides coming up soon too, but this is a picture where we tried to put together where some of these different genes function. And the interesting part is that these are cell types that we knew were involved in lupus to begin with. So again, I think things made sense when we found these genes for the most part. We had a list of genes that worked in some of the immune cells that we think cause lupus. So at the bottom, B cells and T cells. If you've gone to some science-oriented talks on lupus, you've probably heard people talk about B cells and T cells. And some of our therapies, like belimumab, for example, is an anti-B cell therapy, and it works at that stage in the immune system. The big cell at the top is a dendritic cell, and those we think are very important in lupus. They're particularly important in interferons, which the second half of the talk will mention. They're one of the major interferon-producing cells, and there are a number of genetic variations that seem to function in those cells. And then in the dead cellular debris and in the upper left-hand corner, we think that interestingly, some of the things that happens when cells die, it probably makes some of the antigen or the targets of our autoantibodies. So that's something that's somewhat unique about lupus. There's the ANA test, which you've all heard about. If you have lupus or know someone with lupus, you've probably heard them talk about ANA test. That's one of our diagnostic tests. And there are a bunch of other antibodies that often get checked, like the anti-Rho, anti-LA, anti-RMP, and anti-Smith. These antibodies are more specific for lupus, particularly things like anti-double-strand DNA. Interestingly, they can make complexes, meaning that a bunch of these antibodies can stick. In the example of double-strand DNA, they all stick on a long DNA molecule, and they make this big complex of antibodies and DNA that can get taken up inside our cells, and it probably looks like a virus. So a virus is sort of a lump of protein and nucleic acid. It's not a whole cell. A virus is just protein and nucleic acid. Antibodies are the way our normal immune system tags things that are foreign. So if you have a bacterial or viral infection, we try to make antibodies, and the antibodies stick on the outside of the bugs and allow our immune system to know, okay, that's abnormal. That should be taken out of the tissue or the circulation. So in lupus, you make this against things like DNA, which is a pretty common thing floating around the body. And then these complexes, we think, can go inside the cell and turn on the interferon system. And so we think also then these genes that we've found in the interferon pathway, we've generally shown them to be increasers, or things that make the pathway work more strongly, or potentially harder to regulate the pathway and make it more, overabundant. So in some cases, there's a combination of these things that come together, where you get the autoantibodies and immune complexes, someone who has a lot of those, and then the genetic variations where the pathway is turned on. And you can imagine that's one of these multiple hit type processes where, the antibodies cause the signal to be transmitted, and then the genes downstream of that signal are overactive, and then that gets you into trouble. So, okay. So to come back to this idea of prediction of disease, this is a slide that I've used quite a bit for this. It's in a related autoimmune disease, Crohn's disease, which is one of the types of inflammatory bowel disease. But they have had similar results from their genetic screens to what we've had in lupus, where they found a bunch of moderate odds ratio or modest odds ratio polymorphisms that are associated. And this is trying to ask the question, how much can you explain, basically, on the y-axis? And so on the y-axis, if you got up to 1.0, that would indicate like 100%, and you can predict, disease. Each of the dots on the larger graph indicates one of the genes, and so they kind of put them in order, by the number of genes that are found, by the strongest risk factor, the second strongest, third strongest, and it just keeps going as you follow from the green to the lighter blue dots. So you only get up to about 0.2 on the y-axis after all of these 71 genes are kind of stacked up one after another, which would indicate about 20%. You could predict about 20% of what you need to predict disease perfectly. And so that was surprising, I'd say, for most of us, that with 71 risk genes, you still can't predict disease very well. The inset panel, or the smaller graph that's inside the larger one, is interesting in that they thought that these dots came together. It almost looked like a curve. And so on both of the graphs, they have this line that's drawn through them, which is like a curve fit. And if you follow that curve all the way out to 20,000, which would indicate 20,000 risk genes, on the y-axis, you only get up to about 0.5. So that's what the inset panel is showing. And so this actually caused some reaction, as you could imagine, because we have about 20,000 human genes. So that would be saying if you had a risk factor for Crohn's disease in each one of your 20,000 human genes, and if it continued at this pace, then you could maybe be about 50% accurate. And I'll admit, as a geneticist, we got there were some editorials and there were some questions about, why are we doing this? Maybe this doesn't make any sense. Maybe we're never, ever going to get there. That's not true. But there are a number of reasons why that's not true, actually. So I think that's caused quite a stir. There was an editorial in one of the scientific magazine, the case of the missing heritability, and I liked the picture there. It was, where are the genes, and in the cartoon, they show this guy sneaking out the window with the genes. If you look down on the floor there's the ACGTs, dropping out of his sack as he jumps out the window. So what happened, and then of course the shock of the geneticist that we did all these genome scans, you couldn't find it. So I think it's because it's still too early. We have sort of, I think it's similar to like the first few maps of the world or of a country. You have some rough details and the rough edges, but it's going to take a while to fill in all of the specifics. And I think that's still where we are in genetics. So I think one of the reasons that's very particular to lupus, why we haven't found more, and this is what I'll get into a little bit, is that lupus is just not one thing. There's not one path to lupus, and that goes back to our first few slides where we talked about this. That I think it shows up physically differently in different people. It's just there's a lot of heterogeneity there, and so I think likely what that means is that there are probably different genes that are relevant to different people. And when you put everybody together in one bin, say if there's 10% of the lupus patients that have one particular risk gene that's important to them, and then there's 90% of the patients where that gene is irrelevant, you're really going to wash out the effect, right? You put it all together, it's like you kind of average out that 10% with the other 90%, and you don't see very much. So I think that's probably what's happening. And the other one I'll mention is this idea of gene-gene interactions and gene-environment interactions. I think that's really important because none of these things operate in isolation. We know that our genes are in our body and they're constantly part of the environment and they're part of us. And so there must be ways that the genes interact both with each other and with other factors to actually result in disease. I think this is particularly true in complex diseases or polygenic diseases. Monogenic disorders, you probably don't have to think about it so much because you can trace it back to one gene, and when you get that gene, you tend to get the condition. But I think in the complex polygenic diseases, many times they're adult onset diseases, and so it's almost implicit that there must be some environmental trigger or something else. You're born with your genes, and then you get a disease, say you get lupus when you're in your 20s or 30s, so there was this period of time where you didn't have it, and so what changed, what was it? So I think we hold a lot of hope for being able to identify some environmental factors as well. Okay. So to get into interferon, we think that interferon is a causal factor in lupus, and I did want to make that case first. Why would this be important to think about from the genetic perspective? I think there's a lot of evidence that interferon is causal in lupus. And I think one of the more striking ones, that this is something that I saw as a medical student, is that if you give people shots of interferon, sometimes they develop lupus, actually full-blown lupus with kidney inflammation, characteristic autoantibodies, characteristic rash, and it can get better when the interferon is stopped. So interferon is used because it's an antiviral medication, so that makes sense. When we produce it naturally in our bodies, it's an antiviral immune system protein. So it makes sense to use for things like chronic hepatitis. That's a pretty simple concept. Chronic hepatitis is a virus, so if you use an antiviral protein, maybe it'll help, and it does actually. It's an approved treatment for hepatitis. But there are rare people that get lupus when they get interferon, and that's pretty striking because again, we don't really know what causes lupus in all people, but it's interesting that for some people, there's this sort of straw that breaks the camel's back. Interferon is enough to push them all the way into lupus phenotype. So that's interesting. I think in many ways, lupus is at the scene of the crime in a number of different aspects, however we look at it. So the genetics I already mentioned, there are a number of interferon pathway genes actually that are linked to lupus. If you look at interferon levels and when lupus happens, there are higher interferon levels when we're younger, and this is a study where we looked at patients, particularly in lupus patients. You tend to have higher interferon levels in your sort of later teens, 20s, and early 30s. And so that's the lines on this graph are an averaged interferon level across our cohort of hundreds of people. The bar graph is from a different data set, but it's from the Toronto Lupus Group actually has very careful data about when people are diagnosed with lupus. And this corresponds to our general thinking where we think lupus can happen at all times throughout life. That is true for sure, but there's a peak in incidence in the 20s and early 30s. And so think this graph also for me sort of put interferon at the scene of the crime in lupus, because it does tend to spike around the time of onset of disease. And so again, making the case for a causal factor. Most of these studies I've shown so far are a case control design, and I mentioned that briefly. The idea there is that if this inherited factor is more common in people who have lupus, then that should mean that it had something to do with you developing disease. We don't think that lupus goes back and changes your genome, so the arrow doesn't go in that direction. So we really think that the gene somehow led to disease. And in that third bullet, we don't think that the arrow can go both ways. There are some diseases like cancer that cause instability in the genome, and so sometimes that arrow can go the other way. But for autoimmune disease, we find that these are inherited variations that give you risk of disease. So we think it goes from gene to disease. And it makes it useful to study. I'll say the challenge then is figuring out all the different pieces that compose that arrow. Is how does the gene actually do it? And so again, for many things, we're still working on that. And so, the hypothesis is pretty minimal in this kind of study, but I think that can be helpful. It can allow you to discover new things. You don't bias your results from pre-existing ideas. And then I think when we got a lot of hits in the interferon pathway, it gave us an independent level of evidence that interferon is important in the pathogenesis of lupus. And if we looked in families, this is another study that really got me set down this path. You can actually study lupus patients and their healthy relatives, and you find that, as expected, the interferon levels are highest in the patients. But if you look at the healthy relatives, there are often a number of other healthy relatives that have some elevation in their interferon. So this is what we would expect based upon what I've presented so far, is that there are likely some inherited risk factors, some of them in the interferon pathway that turn on the pathway that sort of, are passed down through families. And again, we all have these. So if we all genotyped ourselves for all the interferon-related risk factors in lupus, we probably all have at least one. It'd be unlikely that we don't have at least one of them. But if you have more than average, then that's where we think there's some risk. And then probably the patients are sort of people who got the unlucky inheritance of a bunch of these things that turn on the same pathway. And so healthy unrelated would just be controls, again, healthy people outside the family. And in general, the healthy unrelated people had lower levels. So this, again, supported the idea of a genetic trait, and that this was a genetic phenomenon. And so we've actually gone and tried to map genes using this idea. And I'll say, to get back to the missing heritability slide, it's one of the ways we think we can work on lupus. So not all lupus patients have high interferon. It's somewhere around 50% of lupus patients that have high interferon, up to maybe 60 or 70%, but it's not everyone. So given that we thought interferon was causal, and there was good evidence that interferon was one of the factors that can lead you to lupus, we thought if we could find genes related to interferon, that might be a way to address this heterogeneity problem, because maybe there's a good chunk of patients that have interferon as one of their causal factors, maybe one of their big causal factors. And then if you can study and compare the differences between high interferon and low interferon lupus patients, then you could begin to understand how this differs and how patients differ from each other. And again, that takes us to that idea of personalized medicine. And so, again, many of these things, many of the genes function in the interferon pathway. I think I'm going to actually, let me see. Yes, we're going to mostly talk just a quick picture of IRF5. So with IRF5, we had studied this gene. It's one of the better known lupus risk factors. We found what we expected to find, that if you carried this IRF5 gene variant, you tended to have higher interferon levels in your blood as a lupus patient than if you had the non-risk version. The interesting part is that if you had some lupus-associated autoantibodies, like double-strand DNA autoantibodies or anti-Rho autoantibodies, then you could really see the effect. And if you didn't have the antibodies, you didn't see so much genetic effect. So this is an example of what I'd mentioned earlier, where if you have something that turns on the pathway upstream and then you have the permissive genetics, that might be what has to come together. And I think it's the type of complexity, unfortunately, that makes it so we can't just sort of come out of a gene screen, have all the answers, and be done. I think that's what gave us that graph that kicked off all the editorials where we could only predict 20%. It's complicated, and that it makes sense. Mother Nature had no obligation to make it simple for us, but luckily, I think if we chase down pathways, we can get to a better answer. And so when we looked at how IRF5 contributed to lupus, it was most of this genetic effect was within these antibody subgroups. So it was greater than 70% that was explained by these antibody associations. So so it was a gene that you could get in a general screen and you could see some signal, but you really got a much stronger signal when you knew which group of patients it was relevant to. Okay, and so how this comes together, we think that there are, the changes in our genome, and these genetic changes can come in different versions. So in the pinkish purple dots, I just put gene names, and then the yellow dots, these are different ways that the gene is changed. And then we think they probably work through molecular phenotypes. So the genetic changes then might change like your interferon level, maybe make you more likely to make autoantibodies. And then maybe it's when these things come together that you actually get the physical phenotype. And so again, this is another illustration that if you wanted to go from the pinkish dots genetic variation all the way over to nephritis in the purple dot, it's sort of a complex path, and there are a number of things that have to come together. So this is again why I think the complex disease genetics has taken a little more work, and it's been harder to get to than single gene. Okay, I think I'm going to maybe skip ahead because I want to get into treatment here. I guess I can briefly present this. The idea interferon does play a role in a few different autoimmune diseases. We've done some studies in juvenile myositis that have been interesting in this way because the high interferon disease, and we have a lot of expertise. So we've tried to collaborate with the investigators that work on juvenile myositis and provide our interferon expertise to them. It's interesting because it seems like interferon probably plays a role in a few diseases. When we did a big family history study with a large registry at Northwestern University where they collected family histories over 30 years from some of these juvenile myositis patients. And we found a strong increase in lupus. And so that's maybe hard to see. I don't know if I can point on the graph, but the fourth line down, lupus, if you come over to the estimated risk, 5.89. So it's like a 5-fold increased risk of having lupus in the family if you have someone who has juvenile myositis. And so it's interesting because we think that these are probably both interferon-related diseases. And so again, if this may tie it together for you a little bit better, if you had these genes that cause higher interferon levels, I think when we look at family histories, it's not just that people with lupus, for example, that there's more lupus in the family and nothing else. Unfortunately, many times there's lupus in the family and there may be some other autoimmune disease that runs in the family. Maybe if we understand some of these molecular pathways that are turned on genetically, then you could understand why these things come together. And I think one of the ways we describe this sometimes when describing people's family history to them, and this is again, you don't need any fancy technology for this. This is just from astute clinicians taking history through the years or from large families. You get this sense that there's maybe this antechamber of autoimmunity, what people have termed this, where there's some genes that probably set some kind of a threshold. We think interferon may be one of these that makes you a little more likely to develop autoimmunity in different ways. And then maybe there are some other genes that would, or other environmental factors that push you down one particular path or another. And so in some ways, maybe interferon, puts your family in this autoimmune antechamber, and then maybe lucky family members don't go anywhere. They just stay there. Maybe they have a high interferon level like that family interferon graph, but they don't actually develop an autoimmune disease. And some that are less lucky maybe walk through one of the doors in the antechamber toward, maybe juvenile myositis or lupus or RA rheumatoid arthritis or SS Sjogren's on the graph and other things that are related. So we think this may explain what we see. We've observed this for a long time. It's been interesting that it's not just one autoimmune disease that often runs in families. There tend to be clusters, and so we think this molecular idea may help explain it. Environment, I just have a slide about environment, and that we really don't, it's hard to study it well, but we do think it plays a role. Epstein-Barr virus is one of the things that's been implicated as an environmental factor, and so it's hard to really nail that down, though, because Epstein-Barr virus is a common virus that most of us are exposed to in our life, and luckily most of us don't get lupus, and so there's this disconnect where it means that there must be other factors that are related that we don't know about yet, and how does this all play out? So it's still a frontier, but I think it's an interesting and important one. My talk has mostly been about genetics because I think we've gotten much better at mapping genetics and being able to study it technically. And so we made more progress in that area, but I don't think the environment's unimportant. So I don't want that to be implied as a message that the environment's not important. It's just I think we can study the genetic part a lot better now than we ever could before. One example from rheumatoid arthritis actually is that for environmental factors you can study well, sometimes you can find things. So there's an interesting story in rheumatoid arthritis where if you have one of the strongest genetic risk factors for rheumatoid arthritis and you smoke, you get a much greater risk of developing rheumatoid arthritis than if you don't smoke. So that's the bar graph on the left side of the screen where you see that the bars are basically rising up fairly far from the bottom there. In particular, the reddish bars instead of the blue bars. That's the difference there is smoking versus not smoking, and the higher the bar indicates a higher risk of rheumatoid arthritis. So the genes go from like lowest, medium to highest if you carry 0 copies, 1 copy, or 2 copies of the risk gene. But you can see that one that has the 40 above it with the red bar, that's if you have double copy and you're a smoker. That's a pretty high risk of disease, actually. 40-fold is significant, and it's much greater than if you don't smoke. So I think more of these things are out there. I think that one of the reasons why this study probably was able to find it is that generally people know how much they smoked and for how long, and so you can get actually a pretty good number and amount to put into your equation to get something useful on the other side. I just want to make one last pitch on the complex disease genetics is that we have done a lot of work on this. It's been supported by the Lupus Research Foundation, which Regan is the director of. Regan's going to speak to us next, and she knows this story well, but we've been able to actually come up with some combinations of genes that are highly predictive for lupus. So the current state of the art in lupus is on this slide, and it's similar to that slide I showed from Crohn's disease. We can't really predict very well, but I think we've actually made some breakthroughs recently. I have to sort of hold back on sharing everything because we haven't been able to publish it yet. We're working on publishing it, but it's there's some excitement in this field looking at gene-gene interaction, and we're really excited that for some proportion of patients, it's probably about 10 to 12% of patients, we may be able to be pretty predictive actually with some of these combinations of known risk factors. And so it takes a fairly complex combination, and that's why we haven't been able to find it to date. But we've been actively working on this, and I think it could come to the clinic sometime soon. It's still not something you can get from either your physician or from your 23andMe, but I think that this is something that we may start to shed some light on in the next few years, and so definitely watch this space. Okay, so the last, I know I'm a little bit over the hour, but I guess we got started a little late. I do want to hit a little bit of the idea of therapy, because I think interferon is something we worked on for a long time from the genetic and the science perspective, and so we've been really excited now that drug companies have picked this up and sort of moved forward with trying to block interferon in lupus. And given all the slides that have come before, you think this is probably a reasonable thing to try to block. And there are a number of things that have been worked on. So there were antibodies against interferon alpha. There are a couple different types of interferons that function in a similar way. So interferon alpha and beta are 2 major ones. I'll say that when these were being developed, I was interested in this approach to block interferon alpha because it's probably the one that's more common in the circulation and found in the blood of lupus patients. And it's found in greater amounts in most people, and so I thought that might be a good one to block, and then you could leave interferon beta around, and maybe that would help you fight off viruses. Both of those drugs did not advance, though, out of phase 2. So cifolimumab and rontelizumab were 2 anti-interferon alpha antibodies, and they did not get out of phase 2. Anifrolumab is the one that did make it through phase 3 and now just to approval, blocks both by blocking the receptor. So anifrolumab blocks the whole type 1 interferon receptor. You can't get any signals from alpha or beta, so it's a little more of an aggressive type of an approach. And then there was a group that tried to make a vaccine strategy where you make your own antibodies by being vaccinated against interferon. I don't think that's gone forward, but I haven't heard recently. They did phase 2s, and I'm not sure about what the status is on the phase 3 study. So I may have mentioned some of this already. Both rontelizumab and cifolimumab, the anti-interferon alpha antibodies, showed some initial promise but didn't go forward. Anifrolumab had better results than these in their phase 2. There was a lot of excitement after their phase 2 study. For those of you that followed this closely, the first phase 3 study, which are the big studies that lead up to FDA approval, actually didn't meet the endpoint, and that caused quite a stir in the field. We were a little worried about this then. There were 2 phase 3 studies that were being done in parallel, but the first one to report results did not make the endpoint. The second phase 3 did, and that was a big success. We were very excited about that. And so I think when we look back, it's just that these trials are really hard. I guess the next slide, I'll talk a little bit about those 2 studies. I think the truth is we don't completely know all the details about why one study worked and the other one didn't. The design of both of the studies was pretty similar. I think we still have some degree of unpredictability in lupus trials, unfortunately. But the good news is that it was enough evidence taken all together that it was able to support an FDA approval for the drug. And why I say that's good news is I think we still just need more tools in the toolbox. It's not a miracle cure for lupus. We all wish that something would be, but it looks like it will be useful for some patients. The acronym for the trial was TULIP, so the Tale of Two Tulips studies. There's one endpoint that was met in both trials, and so, but it wasn't the reason for the difference actually necessarily in the outcomes. I mean, the fact that this SRI endpoint was not met in the first trial, it actually just looked like the results for the drug were better for both endpoints in TULIP 2, and so it's really not clear why. We do have difficulties in lupus trials. There's a high placebo rate. There's a lot of background therapy, and some of the things we talked about first with all the undulating disease flares and remissions, it's hard to track that honestly in a clinical trial and to really assign what's due to the drug and what's due to who knows what else, other things driving flares and remissions. So, and the last piece I think that's in the picture, and I want to just sort of end here, is that I think we're still a little ways from personalized medicine, unfortunately. So this graph is from the phase 2 study, and it was exciting in some ways because this, all patients is just the results. The 2 colored lines are the active intervention group that got the drug. The gray line is the people who didn't get the drug, and it's the response rate. And so it's about a 40-50% response rate in people who got drug overall versus more like a 20-30% of people who didn't get drugs. So it's a sizable difference. And then if you looked at the interferon high panel in the middle, it looks like there was maybe even a little better response and a bigger difference and less of a difference in the interferon low group, although the lines look a little noisier in that interferon low group, and that's because they didn't put as many subjects in that arm. And the other part that was a problem is that the placebo rate, or the gray line, actually was higher in that group. And so we got excited wondering if this might mean that people with high interferon levels might respond better to anifrolumab, and if so, maybe there'd be a biomarker and a way to personalize treatment. The phase 3 data didn't really strongly support the interferon signature as being something that's predictive. And so I think this is beyond this talk, but I think the way the interferon signatures are done are probably not specific enough, and we may have to just get a little bit smarter. May have to work in some things like genetics and some of these other things, because it's, I guess one of my take homes from the first part is that it is complicated, and it probably is not as simple as just an interferon signature in deciding who can get which or another drug. So, so to wrap up, I think there's a lot of encouraging news lately. So I think that, genetic mapping in some of our early biological studies encouraged type 1 interferon as a potential therapeutic target. And I think similar to belimumab, the importance of B cells in lupus encouraged B cell target development. And we've had drugs now that actually achieve FDA approval for these specific targets. I think that's great news for us in the lupus community. I think we're going to need a combination of things to really map out how patients differ from each other. If it was simple, we'd probably already be there, but I think that we have to have probably a relatively comprehensive way to evaluate people's disease, including both genetics and immunological markers. If we can do that, I hope we can enable real personalized medicine, and that's still my hope that we can get there, and the sooner the better. And I think this is a major milestone. We've had a few different approvals actually recently, and it's really an exciting time, I think, in lupus in general. So thank all the members in the laboratory who do this day in, day out to try to help us make progress. The Lupus Research Foundation and the other funders, really thanks for your support, because you really helped us get there. And to keep all this moving, we just try to make sure we have enough to cover everybody's salaries and keep everything moving, and all this support from donors and foundations and from the Lupus Research Foundation is really helpful. And thank you. I should stop there and take any questions, Chad.
Great. Thank you, Dr. Niewold. That was, gosh, so informative, so much information. We've got a couple questions that have come through already. And remember, participants, you can type your questions anytime now, and we will ask them to Dr. Niewold. If we run out of time, we will send a list to him, and if he has a chance, he'll answer those to us via email. Okay, so feel free to start asking your questions. Our first question is, in your opinion, what are the most promising lupus medications in development?
It's a good question, and I know it's one that I'll say probably my colleagues in the pharma companies ask themselves every day when they get out of bed, and unfortunately it is a tricky one to answer. I'm not trying to cop out on the question, but it's I think we have a lot of unexpected things that happen in drug development, and this is we very much in the royal sense because it's not what I do as an academician, but it's hard. I think just the last example of the anti-interferon drugs, if you would have asked me on that picture slide which ones I think are going to be most likely to make it and have the best safety profile and impact disease, I picked wrong, and I studied the pathway, and so there's a lot of mystery to this still too. I think I would have guessed that maybe anifrolumab might have been aggressive, honestly. We have interferons around. Interferon is one of the things that helps us fight off COVID. There have been a number of papers suggesting how important interferon is actually in our defense against COVID. And so when these general concepts were pitched to us in the research community about which way would you go at it, we heard anifrolumab, and I wasn't alone in thinking, gosh, this is something that's been with us forever in our immune system. Do we really want to shut off the whole receptor, all of it? I don't know, maybe not. And now it turns out safety profile, it looks good, everything has its issues, but I think it looks good, and it actually is the one that made it and was efficacious enough in the trials to make it. So it's hard. I'll say, so I don't sort of, hopefully I can add maybe one more facet so hopefully answer your question. I think interesting treatments, I like those that are directed at the pathways that we know. So I think pathways, I have my own bias toward interferon, but I think B-cell pathways, there's a lot of interest. I think T-cells are really important in lupus too. I think because of the antibodies that we've known about for forever, and the fact that the B-cells are the part of the immune system that leads to making antibodies, we've had kind of probably a B-cell bias in lupus in some ways, but it worked. We got belimumab across the line, so that's great, but I think there's probably still some room to think about T-cell directed therapies because it's clear that T-cells are important in lupus as well. So I don't want to, name any names specifically about drugs, but I think that some of those pathways will be good to go for. And we just have to keep pushing, I think. The better we can define what goes wrong, the better we'll be able to target it.
Great. Here's another question. Dr. Niewold, my husband's family has suffered from lupus for 3 generations, going back to my husband's grandmother. Is it possible lupus could skip a generation in the children from the 3rd generation? This family also has 2 members who have Crohn's disease.
Yeah, so that's a great question. And skipping generations is pretty common, actually. And so if you have like a whole sort of a family diagram, it's not uncommon to have maybe a more remote relative who's had lupus and then have it come up again later. That's some of the complexity. You could imagine if you had, 20 some lupus risk genes and each one of them sort of gets passed, with like I said, like kind of like a coin flip, maybe gets passed, maybe doesn't to each person. There are a lot of different combinations that will be present in the family. And then I think the other part of that is the environmental exposures, which again, we don't really know, unfortunately, a lot of the environmental exposures, but there should be some. And so there's probably some bad luck component to it as well. One of the interesting things is that our more distant relatives, we actually become much less related to them. So a twin, you're 100% related to, so that's your most close relative you could have. Your other relatives, you're only about half related to, again, because you get half your genes from your mom and half from your dad. So if you have 2 unrelated parents and they have children, each of your each of your siblings, you're only going to share about half of your genetic material with. So in some cases, some of those families, the reason why I say that, some of the family histories, it could be that there was a confluence of lupus risk genes, and maybe then it sort of drops away because the next person who marries into the family doesn't have so many lupus risk genes, and then maybe the next time, the person comes who brings in maybe some different lupus risk genes, and so again, we're not smart enough to track all that and know how it all happens. So it's, but that's another possibility that could be, because, when you go by halves, you're only like 1/8 related to more distant relatives or 1/16, and by that point, it's unlikely that you just happen to get all of those lupus risk genes from that person. So yeah, it's a great question, and having Crohn's or other autoimmune diseases, we do think that some of these pathway genes, again, interferon is the example that was presented, that it probably leads to a few different autoimmune diseases, and that might be sort of some part of this explanation of why we've observed this for a long time. It seems like autoimmunity can run in families, sometimes the same condition and sometimes not.
Okay, great. Here's another question from another lupus patient. I grew up in a farming country, in farming country, and my sister and I have lupus, as well as a cousin a block away. There were also 2 siblings down the same street who developed lupus. How much part do you think pesticides and a power plant might have played in this?
Yeah, these are really good questions, and I think both of these, we've tried to do some studies. I'd say it's not as definitive as I'd like, but I think they're both potentials that we're thinking about. I think environmental pollutants have been a little bit tricky because I think we haven't always had great data. And it seems, I think it's a shame, there are some organizations that try to do this, there are some organizations that try to collect this information, but as a researcher, I'll say when we've tried to access and see what's there and what can we look at that's really well measured, quantitative, you can hang your hat on it, it's not so much, unfortunately, but I'll say that we've had, historical evidence that would suggest both of these exposures that you list could be related to your risk of lupus. And so I think we are luckily at a point now where there are some researchers who are digging in deeper and trying to understand this, because I think it's, we do a lot of things, as humans, we've changed the world quite a bit from how it was when we evolved, and we've done a lot of things are different. I think if humans from, thousands of years ago saw us now, they'd think, what happened? This is totally different. So I worry about it. For you've developed lupus, and I'm sorry about that, but I think that now obviously the best thing is thinking about how to treat it and handle what's there. It's still a complex thing. So, we don't have anything that we know of where you can sort of walk into a room and get lupus either. So it's always, I think that's also hindered our studies in some ways. There's probably people who have some background susceptibility, which again, you wouldn't know about maybe, and then the right environmental factor comes along, and then that's when it happens. So it has also made it a little harder because it's not, we don't get really clean data where there's some exposure and then everybody around it gets lupus. It'd be a tragedy, but of course it would also implicate that factor very cleanly, and you would know. I think some of the mystery and why this has been so elusive is that it probably also takes the right person who's kind of set up with their immune system in a particular way. But we'll get there, and I think it's one of those things that I hope that pesticide use will become more biologically friendly as well. I think that's something that we probably still need to work on, and things like where we locate, some of these industrial sites. I think that through our history, at least in the U.S., we haven't always been very cautious with that, and probably could use some more caution.
Yeah. There's another question. After the talk about interferon and immunity, what is the recommendation for the COVID-19 vaccine, and which one?
Yeah, so it is, we've done some studies. I guess I'll start off with some of the data we've collected so far in the pandemic. It looks like the risk factors for getting severe COVID, at least, that would bring you into the hospital, looks like a lot of the same ones in the lupus population as the general population, which is good news. It doesn't look like lupus is such a risk factor necessarily in and of itself. It looks like and so what I mean there is like hypertension is a risk factor, and having a higher BMI is a risk factor for more severe COVID. Lupus doesn't eliminate those, but lupus by itself isn't a major risk factor. It's more that if you had lupus and then had another risk factor, then that risk factor would be active. Corticosteroids, it seems like, or steroids. If you're using steroids at a solid level, there's some risk with that. And the other medications, we haven't seen clear risk signals yet. As you know, there was this whole hydroxychloroquine story. It looks like that's probably not a major factor either way. And thank goodness, I think mostly our patients are all getting their hydroxychloroquine again, and the rush is over, thank goodness, because it is really good. We know it's really good for lupus. With COVID, it didn't seem like it did very much. So that's sort of the background on your risk. There's still the risk. It's just one of these new bugs, and we don't have defenses against it. So that's been why it's just sort of ripped through the human population the way that it has. Vaccination is recommended. There have been studies about, this general question in the past, do vaccines tend to clear your lupus? And we don't see evidence of that. It's a fair question because the vaccine stimulates the immune system. And so it's a good question to have because you can wonder, in general, we haven't seen population level data that suggests that there's a large risk to the general lupus population getting vaccines. That being said, I know people who have had flare-ups after vaccination, and so again, it's, if that's you, then you'd say, okay, Dr. Niewold, but it happened to me, so come on, and so that's the hard part, is that population data, is important to you in some ways, but if you're the N of one, then that's the most important thing for you. But in general, the safety profile of getting the vaccine and how COVID has really, it's been such a dangerous virus for so many people, we're recommending that you get vaccinated, especially the fact that if you would have to take steroids or have some other immunosuppression, you'd be at an increased risk of having a more severe case of COVID. And so if you had antibodies from the vaccine that can help protect you, then that would be a good thing. So we've been recommending get vaccinated. Your immunocompromised status will usually get you, everybody will want to vaccinate you. And now the vaccine is very widely available. But in early days where we had more limited numbers, we were able to get our patients vaccinated generally a little bit faster because they were immunocompromised in some way if they're taking medications. So yeah, great question, very topical. We wondered if the interferon present in lupus, I don't know if this is part of your question, if the interferon present in lupus might actually offer some protection. We don't know, but we're actually trying to do some studies in that way. If so, it's not going to be a major thing. And again, get your vaccine. Lupus isn't going to protect you from COVID, but it's an interesting question. I think from the more of the genetics and the basic science question, I think there's so far it looks interesting. We have to push into it more, but that would it could prove one of these principles again from the theoretical side, that some of these things that give us risk of lupus may actually be a benefit to us in our immune system in some ways as well. And they're not necessarily bad genes. It's just that you get sort of an unlucky combination. So, but again, that's not a reason to not get vaccinated, and do please.
Right. Going back to the genetic question, this patient has a strong history of autoimmune diseases on both sides of the family. However, they're the only one with lupus. Do you have an insight about how the family of diseases manifests?
Yeah, so let me make sure I get the question. So many people with their family history, you may not find lupus at all. So that's actually one of the more common family histories. If you meet lupus patients in the clinic and you take a family history like we all do, it's actually reasonably common to not have anybody else affected by lupus. Another thing that's common is you could have people who could have positive blood tests. And so some of you on the call may have had this experience, and then that's scary, of course. But there's an increased rate of having some of the blood tests be positive without developing lupus. And so I hope some of the talk may give you some ideas about that, that some of these molecular features maybe get inherited, but maybe not enough to get the whole thing. And so that's actually not too uncommon when we look at families and study them. We can find that, for example, the ANA test is not uncommon in relatives. And those relatives often are fine their whole life and they never get lupus. So again, that's obviously scary. If if one of your relatives has lupus and then you get a positive ANA test, that's going to shake you up. And I understand that fully, but there's some reason to feel somewhat reassured. We don't have a prevention, unfortunately, but we usually will follow people closely and just make sure everything's good. But generally, my advice also is if we don't find anything besides the ANA, my strongest advice is please don't lose a lot of sleep and carry around chronic anxiety from that, because you'll probably be okay. So if there's a way for you to sort of put it out of your head and just see me every once in a while and get checked, and then hopefully get on with your life, because it'd be a shame to worry about something your whole life when you're not actually going to get it. So that's some of the things that can happen that are sort of like lupus, or sometimes people can develop an incomplete lupus. Some of you on the call may have had experience with that too. Lupus is not well understood, and some people will have symptoms that fit with lupus, but maybe not the full-on, these formal criteria. And if you meet 4 of 11 criteria, you can be diagnosed with lupus. We have a bunch of 3s. There are a bunch of 3s in the world, and it's interesting. It's something. It's not nothing. There's something there. And I think that's, again, because Mother Nature doesn't care whether we say 4 or 3. It just does, she does what she does. And there's a spectrum. Most things are a spectrum. Lupus definitely is. So that happens in families too. And I think that's the same thing. It's maybe instead of just having positive blood tests, maybe somebody has positive blood test and a rash. And, and luckily, again, maybe they didn't get enough of the other parts of the puzzle to really develop full-blown lupus. We're always vigilant. My hope is that maybe some of the genetic testing that we can do as we get better and better at it, you might be able to get a more rational sense for people who would want to know. There's a lot of personal variation in that. Some people really don't want to know, and that's fine. But for people who said, I want to know everything I can know, my hope is that we would have some genetic testing we could do that could give you a better sense. And then that would be great, because most often the answer would be more reassuring than what you would think, because again, lupus runs in families, but not that tightly. So most relatives of lupus patients will not get lupus, and so if we could have some genetic testing that could add some additional confidence to that, it might actually be really helpful in sort of allaying people's fears. So, or if it came out the other way, at least to be vigilant. And if we get preventive strategies, there might be a better way to implement those, because you would know who's really at risk, who do we really worry about, and then maybe there would be things we could do for that subset.
Going back to your genetic testing and your talk about the grouping of genes, and I know you said that it's not available in clinics yet to patients, but do you kind of have an idea of, is this near future, far future that this would happen? Or
I would hope in the next year or two we could get something moving. It's a little bit hard to predict, but I think the technology there is a good part. So I think that these are things where the information content is mostly what we have to figure out and how to implement on a broader scale. So I'm excited. I think there may be some things, and in particular, this type of testing would probably be most useful in this scenario that we just talked about, and it's sort of a high-risk category where there's some reason why you're worried about your risk of lupus, and then you could check this and see if you carried this particular combination of variants, then you'd be much more concerned. And if you didn't, then that would be a good thing. So I think there could be that could be fairly soon. I'm excited about that. We've been working through things like IP in ways that you can sort of make sure it can get out into the real world. And the reason it's not tomorrow is there is like sort of this translation gap trying to get basic science findings into the real world. Unfortunately, there's all the pragmatic things that you have to do, even simple things like how would labs that currently do testing actually do the testing and make it work. But it won't be forever. So that I'm excited about that. And my hope is that we'll continue to sort of break down the puzzle in different ways. I think this is an early success that'll be some particular group of patients this will be useful for. And then my hope is we'll get to the other 80–90% and just sort of keep chipping away until we can get better answers. So I think that would be good. Again, if you've already got lupus, then it's not as helpful. It's like, asking for the weather forecast now when you can look out the window and it's raining. It's not as useful, you sort of already know. But it may be helpful. I shouldn't be that negative about it though, because it may indicate some type of disease or some particular treatment that might be helpful. So that's the other thing that I think is hopeful about genetics, is that even if you already have the condition, it may say something about the wiring of your immune system, and it might be able to suggest which treatments might work better for you as compared to someone else, because you know which road you travel to get to lupus in the first place.
Right, great. Let's see, one more question. We're kind of slowing down on questions, so if anybody has a question they'd like to ask, please type it in now. This one, what about your impressions of the potential future findings based on the trajectory of current research?
I think it's an exciting time. I think that in general, immunology continues to grow as a field, and that's one of the the fields in biology that really supports our research in autoimmune disease, and that continues to get more sophisticated, more exciting. The technological advances have been really exciting too. So I think that that's some of the parts of science that are really moving along. We've started to incorporate some things like machine learning into our own research, for example, to try to get at some of these complex patterns. As I mentioned in the talk, if these things were simple enough, we would have been able to say, okay, we looked at X, Y, and Z, and it's Y, but it looks like it's going to be nothing like that. It looks like it's going to be the square root of X plus, the logarithm of Y times something else. And, these things that you can't just sort of just come up with, just look at something quickly and come up with. But, if you had like a computer cranking away at some of these things, and running down all these iterations, maybe you could. So, and there's a lot of art to it. It's not that simple, obviously. And for anybody who's into machine learning, it's not that simple. It takes both of us. It takes our biased human mind and it takes the unbiased computer mind, so they're working together in a good way. And then you can start to crack some of these things. But we're excited. So I think that's some of the substrate that'll help us. And then I think better understanding the immune system. I'm really hopeful for new targets. I think will continue to emerge, that we'll get better ideas of things that we can, where is the best button to push to really change the immune system. I think my feeling is we're backing into it. We're getting some major pathways. For example, to try to maybe make an example from the interferon pathway, we blocked the receptor, which is a pretty basic strategy. It's a very big turnoff. Like I said, I was actually kind of worried about it, whether that would be safe, but that's been helpful. But I think then if you could get more specific, and if you knew, well, this is where the pathway is really turned on, and then you could go after this particular target, the benefit there could be in side effects. Again, we don't know until we get there, but it might, my sense is the more graceful you can be with therapy, the better it'll come out, and probably the more effective too, that you really hit the right spot. So I think in general, drug development, we keep pushing in this direction, and I think we sort of gradually get there. So I'm hopeful that these would be some of the things on the research side as we understand disease better, both genetics, immunology, put it together, and then hopefully identify new targets, or from even existing things in the armamentarium, know which patients really need which thing. I think that's the other major frontier that I'll end with, is just for patients and myself as a physician, it's humbling and it's rough, honestly, because we try to pick the best first therapy, but as many of you patients know, it's sometimes the first thing picked doesn't work, and then you try the second thing, and then it's really frustrating for everyone to have this somewhat of a trial and error strategy. I mean, we use all the information we have at hand. We try our best, but our best is not good enough. And that's actually why a lot of my life I've dedicated to research, because I feel like we have a ways to go, and it's frustrating in the clinic. I know it is. There are a lot of treatments you try and it doesn't work, and sometimes you even have side effects, and then it's really, I think, gosh, why did I do this? It didn't help me, and get side effects. So that's the other way I think, though, that I'm hoping science will advance us, is to know what's the right drug for the right person, and that sort of ideal of precision medicine. I hope we can continue to make progress in that.
Great. Well, thank you, Dr. Niewold. I think that's a great place to end, and a hopeful look into the future. But thank you so much for your time. That was such an interesting topic, and I know that all the patients really enjoyed it, and everybody who was here to listen. Participants, don't forget to complete the survey at the end. This is the end of session 1, and I hope you'll join us at 1 p.m. for our wellness session. Be sure to have a towel and some water, and we'll listen to Regan Burr as she talks about her lupus journey and leads us through some great exercises as well. But thank you again. Remember, you can use the same link to join us, and we will see you soon, hopefully. Thank you, Dr. Niewold. That was great. Okay, bye-bye.
Thanks for having me.
Thank you. Okay.